Thank you very much. Thank you for being here. So I'm going to tell you about trying to explain to you how this little animal is able to swim towards a light source. So we're going to try to understand this process both from a behavioral point of view, so just by looking at the fish actually doing this task, and then we're going to dive into the brain and try to understand how the circuitry of the brain is able to process the visual information and produce this sort of behavior. So you probably may ask yourself, why would you be interested in such a small and tiny little animal? And it's a very practical reason. So the zebrafish has been developed originally for development biology, because you can look at the first cell up to the complete animal pretty much under a microscope, and that takes of the order of a few days. But recently it's also been very useful for people doing calcium imaging. So calcium imaging is a way to record neuronal activity using light. And to do that, what you need is some sort of calcium sensor, so molecules which are fluorescent, but whose fluorescence is going to change with the amount of calcium. So if you have such molecules inside neurons, each time you have a burst of activity, a spike, you can have an increase in the intracellular calcium concentration, and that is going to be converted into an increase in the fluorescence that you see. And because this animal is so small and is also very transparent, you can actually see the whole brain here when you have included this thing genetically, right? So, and because of this very nice property of this animal, in 2012, people were able to produce the first whole brain activity mapping of a vertebrate brain. So the idea here is that what the color here shows the population of neurons that activate upon a certain sensory stimulation. But to do that, also the brain is small. It's only 100,000 neurons, and it's about millimeter in size. To do that, the people here had to take of the order of 30 to 50 different brains, do an imaging in a little part of the brain, and then fuse all these recording together on some sort of reference brain. And the reason why they had to do that is because they were using a microscopy technique called two-photon microscopy, which is intrinsically slow. And so something I'm going to tell you about also is this technique of microscopy that pretty much at the same time started to gain a lot of traction in fluorescence microscopy. So in this technique called light sheet imaging, what you do is you project a thin sheet of light across the sample, and you record the fluorescence at 90 degrees compared to the light sheet. And when you do that, you can do very long recording with very little photo bleaching. And that's the reason why, for instance, this technique proved to be really useful to look at the embryo development for many, many hours. So there were people here who were able to actually follow the motion of many, many individual cells for hours and hours, something you couldn't do with confocal microscopy and two-photon microscopy. So I'm going to just take a few minutes to explain to you, just to give you an idea why light sheet microscopy might be really useful in the sort of thing that we do. So when you try to image fluorescence within the sample, within a volumetric sample, the problem that you have is that you want to image this plane where you have fluorophore, but you don't want to have signal coming from the other part of the sample. And so to do that, what people invented about 50 years ago, it's called confocal microscopy, and the idea was to put a little pinhole conjugated from this point here, so that any fluorescence that comes from above or below will be stopped. And so essentially, the only fluorescence that you get from this little spot here, right at the focal plane, at the focal point of your objective. And now, if you want to produce an image, what you do is just scan across the sample, and then you can produce the whole image point by point. But you can see that by doing that, to produce an image with n points, let's say 1 million points, you have to to do that again and again. And of course, there's a little limiting time that you have to spend at each single spot because you want to be able to record at least a few fluorescent photons, right? And so that gives you this relation between the acquisition rate, the number of points in your image, and the inverse of this time scale. And that makes this technique pretty slow. And it's the same for the other technique that you may have heard about, which is tau-photon microscopy, which is also a point scanning microscopy technique. Another issue that you have with this technique is the fact that if you want not to record the whole volume, you're going to have to move the plane up and down, right? But each time you're recording one single plane, you shine light also on the top and bottom of the plane that you're actually looking at. So the photonic efficiency is pretty bad, which means that if you want to record 10 slices, each position in your sample is going to take to experience 10 times more light that you really need, because in fact 90% of the time the light is not used for that; it's going to be blocked by the pinhole here. So Lightsheet has really a very simple way to deal with this problem of what we call optical sectioning, which is to say, okay, we're not going to shine line from the top, we're just going to shine line from the side, and so we're going to illuminate the whole plane together. And then you can just project that onto a camera, and then you have a complete parallelization of your whole plane at once, right? And so now your equation becomes 1 over tau dwell is just the frequency of acquisition. So you gain typically 2 or 3 orders of magnitude in terms of the speed at which you can record. And of course, at the same time, when you record this plane, you don't shine light above or below. So you have the maximum efficiency in terms of optical load that you put on your sample. And so that makes it, this part makes it really nice when you want to do chronic imaging, like the movie I showed you before. And this fast acquisition is going to be very important for us, because what we want to do is we want to record the whole brain really fast. Okay, so now this is what you get when you put one of these little zebrafish larvae under this sort of microscope. So this is just one slice. And the change in fluorescence with this green changing in color is the green reporter, and it tells you where the activity takes place. And you can see that at 20 Hertz, we can record pretty much across the whole brain, from the spinal cord here to the telencephaly here, to the midbrain and hindbrain, and here you have a little bit of the cerebellum. So you have a complete view across the entire brain, and here you have of the order of 5,000 individual neurons. Okay, so now once you have that, of course you won't do it by hand. What you're going to do is you're going to zoom in. We're going to take one of these little. So you see these neurons here. In fact, there is no reporter in the nucleus, so you see activity right around it. And if I look at the activity, the fluorescence within this region, you can see this typical burst of fluorescence that is just the signature of the sudden activation of this one neuron. And then you're going to do that over the entire set of neurons, and that allows you to produce a lot of these different time series for each individual neurons, in that case 5,000 neurons. So you can do that at 20 Hertz, which is already pretty fast. It's actually so fast that it's even too fast compared to the intrinsic dynamics of the reporter. So what you can do also is use all this speed instead to do a 3D. So what you're going to do is you're going to sequentially image different plane and then go back and do that again and again. And so if you do that, at the end, you can produce this image, which is a 3D image of the order of 70 or 80,000 neurons in time. And you can do that today at 2 or 3 years for the entire brain. So every 300 milliseconds, you have the activity of 70 or 80,000 neurons. Okay, so what you get is, people tend to say it's whole brain recording at single cell resolution. I would say it's quasi-whole brain. You already have a certain region, in particular between the eyes, where the light can go. And it's not completely single cell resolution, because you've seen that the cells are really touching together, so it's really hard to completely separate the signal. But you have some sort of entire recording of the brain with something which is close to the single cell resolution. Okay, so we've been working with that for the last 5-6 years. Now what you can do, of course, is take this little fish, do this recording, and at the same time, you can stimulate the fish. So we try to stimulate with the auditory system, the thermal system, the visual system, and the chemical receptors. And this is one example, for instance, for the where you see the thermal response, so the response to cold and hot puff of water. And this is just the map, the functional map of the entire brain, where the red regions corresponds to the neurons that respond to hot water, and the blue region to cold water. So you can pretty easily get this sort of entire responsive map. Okay, so I'm not going to talk about that, but I just want to show you this recent project that my colleague Volker Bormuth, who works just the next door, develops in the lab. So he wanted to address another sensory system, which is the vestibular system. So it's the inner ear system. So it's the one that you excite by moving around. So to do that, the problem is he had to actually move the fish around, right? And so he had this crazy idea of actually mounting the entire microscope on a big motor, and then moving around the fish and the microscope at the same time. And so what you see here is the microscope. This is 50 centimeters by 50 centimeters. This is the fish that you see from the front. And we freed the eyes, so the eyes can move. And when you do that, you know that the eyes compensate for the motion, and you can see that the animal actually compensates. And at the same time, you can see 4 different brain sections where you see the activity that is evoked by vestibular stimulation. So you see the activation of the different region, and here at the same time, you can see the eyes compensating for the motion of the microscope and the fish. So I think that's pretty cool. Anyway, we're not going to talk about that. Yes?
I have a question. If you express that much fluorescent protein in brain cells or in neurons, does this influence their behavior in some way? Or is there, if they have so much more stuff in there, that there's no other function for it?
There's probably 2 issues. One issue is, does the protein that is the calcium reporter produce some sort of phenotype perturbation? And the other question is, does the fact that these proteins are going to capture the calcium is going to change the calcium signaling? So that's an issue that I don't think people have ever seen anything that's significant from the phenotype point of view in zebrafish. They've seen that in C. elegans. So at a certain point, if it's too efficient, then it's the calcium signaling problem that gets in. And so that's the limitation. And in fact, I mean, it's a very first principle problem. If you want to measure something, you are always going to perturb it. So if you want to measure the calcium concentration within the cell, you're going to have to modify the calcium concentration in a way.
What's the affinity of the reporter?
I don't have that in mind. But I think... So I think the question is what fraction of the free calcium will be. I think it's on the order of 10% or something like that. Okay, so now I'm going to move to the actual topic of my talk, which is phototaxis. So phototaxis is just this innate behavior of this fish. We tend to go to where the light is. So this is a pretty old experiment in which you just shine light at 2 different positions in a petri dish in which you have a certain number of larvae. And you can see that the larvae tend to navigate toward the light or move away from the dark part of the dish. So what we want to understand is 2 things. We want to understand what is the algorithm underlying that. So it's a very simple animal. So when I'm talking about the algorithm, which is what is the basic sensorimotor response function that is going to produce this sort of overall behavior of phototaxis. And then we're going to try to probe the sensorimotor computation, which means how the brain itself is able to integrate sensory information, visual information, and convert that into the adequate motor response in order to produce this operation. Okay, so there's essentially two algorithms that you can think of. The first one is to say, okay, the fish at a certain point in time, if the light comes from here, is going to see a little bit more light on the left than on the right. So one very easy way to do phototaxis is to say, in that case, I'm just going to turn toward wherever I have more light, right? In that case, to the left. So what I'm going to do is just an intensity comparison between the two eyes, and this is what we're going to call stereo-visual comparison. But in fact, the animal could do something else. Imagine that it has only one eye, so it wouldn't be able to do that. Then what it can do is move somewhere randomly, and then if you move to a certain direction, you're going to see a change in the total intensity, right? And so it's going to sample the gradient of light by actually actively moving forward or turning around, right? And so by doing that, it might be also able to find to infer the position or the orientation of the light source. So we're going to look at these two processes separately. So to do that, we design an experiment in which we have one single zebrafish, which is going to swim, and we're going to project on the left and on the right a uniform, homogeneous illumination with intensity IL and IR. And what I'm going to do is real-time tracking and visual simulation, which means that I'm going to, at each time, every 10 milliseconds, if the fish, for instance, turn around, the projection is going to follow the fish, right? So by doing that, I can impose whatever stimulation I want to the fish exactly, right? So I can completely control the stimulus at any time. And so then we're going to do two things. The first thing is we're going to test the stereo-visual stimulation. So at each time, the animal is going to have a certain contrast between the two, right, which is defined by C. But the sum of the two is going to be constant. So the total illumination is the same, but the contrast is modulated by the orientation of the fish. In the second case, I'm going to shine exactly the same light on both eyes, but now the intensity of the light is going to change with the direction of the fish. And so by doing that, I can test both strategies completely separately. I can separate the two. Okay, so what happens? So the first thing we're going to look at, and I think that's important to understand, is that when you look at the fish doing phototaxis in the real life, it's not completely obvious what it's doing. Because essentially, they don't need to be in the light to survive, right? They tend to go closer to the light, but essentially what you're going to do is you're going to bias this sort of random walk that the animal does. So this is a trajectory of a fish with no gradient of illumination. Okay, so we're going to start from that. The first thing we want to describe is this random walk. So one thing that is important to see, and which is going to make things easier, is that if you look at this motion, it's very jerky. So essentially, the fish is pretty much immobile for 90% of the time, and every 1 or 2 seconds is going to make a very short movement, which we call a bout. So what that means is that the orientational trajectory that we're going to be interested in, so we're going to look at all the fish reoriented over time, is essentially discrete. And because the stimulus is locked onto the position of the fish, the stimulus itself is discrete. So we are brought back to a complete discrete problem of an animal moving around, but by discrete bars. So what we want to have is a description of the statistics, the probability of a certain reorientation, which I'm going to call dx, which is the derivative of this. So is the amount of reorientation for a certain bout. And I want to understand how this depends on what is done before. So what was the bouts that it triggered before, and what is the visual stimuli that it received before this bout, right? So I know I have this kind of function, and I want to understand this function. Okay, so the first thing is we start with the spontaneous activity. So we would just look at the fish without any visual stimulation, or it's completely uniform. So what you can see is that essentially you have 2 types of bouts. So this is the amount of reorientation for different bouts. So what you see is that you have essentially 2 Gaussian, one very wide Gaussian and one very peak Gaussian. So what does that mean? That means that the fish has essentially two ways of moving. Either it is moving straight, so it picks from this distribution, or it's turning around, and it picks from this wide distribution, right? So it has to choose between turning and going forward at each time step. So things will be easy, except that it has a little bit of memory. And so if you look at the autocorrelation of this reorientation, dx, you see that it doesn't go down to 0 immediately. So that means that, and in fact, we can describe that by a first-order autoregressive process, which means that essentially, if you know the bat that is down before, then you know the probability of the next bat. And what happens is that if it turns to the left at a certain time, then the next bout, if it turns, has 80% chance of being to the left, right? So there is this parameter which I'm going to call Q flip, which is the probability of changing direction. And this probability is not 0.5, so it's not completely stochastic, but it has a large probability of going the same direction. So essentially for 4 or 5 times it's going to go to the left, and then 4 or 5 times it's going to go to the right. Okay, so then you can describe it this way. So you either turn or go forward, and if you turn, either you turn in the same direction as before, or you flip direction, right? And then you pick from the left or the right. This is like the easiest way to describe these trajectories. Okay, so that means that somewhere in the brain, you must have some sort of system that keeps tracks of left or right turning. So I'm going to call that some sort of state, a left state or a right state. And the system oscillates from left to right, and it switches from one to the other with this sort of telegraph signal. So you stay for a little while, 4 or 5 times, and then you go down to the other state. Okay, so now, first thing, what happens if we put some sort of stereovisual stimulation? So we impose a contrast, and so in that case, what we do, so this is 0 and 2 pi. So this is the direction of the fish. So you can think of that as the fish pointing to the north, the fish pointing to the south. And so what I'm going to do is that when the fish points to the north, it has exactly the same intensity on both eyes. If the fish points to the east, then it has a lot of intensity on the left and very little intensity on the right, and so forth. So if I look at the contrast, which is the difference between the two, it looks like that. And actually, this is the one stable point. If the animal goes to the light, this is the one stable point. So this point is unstable, which means that if it turns a little bit, then it has to bring it to the center. So essentially, the animal, we expect the animal to go towards pi. And so this is for one fish, different trajectories. And as you can see, in that case, at least, it does a pretty good job of actually going to the right orientation. Okay? Now we can do that for many, many different fish. So I think, this is like 40 fish, so a lot of trajectories. And you can see that in average, the animal tend to point toward the light source, just by having this comparison between the right and left eyes. Okay, so now what we can do is, because we want to understand this difference, what we can do is just look at the average reorientation versus the contrast. And it's essentially linear, which means that given a certain contrast, the probability of turning left versus right is just proportional to the contrast itself. Okay. But in fact, what you can see here is that, remember, you have these two distributions. Essentially, the forward distribution doesn't change, which means that when the animal goes forward, it really goes forward. Doesn't care about where the light comes from. But whenever it turns, then it has this linear relationship. And you can see that the slope is twice here, simply because half the time it just doesn't turn, right? So this effect comes from the average of these two. So we have a left-right contrast bias, the ratio of left versus right turns. And this contrast doesn't affect the ratio between forward or turning. So the animal doesn't turn more because it has some large contrast, it turns half of the time. But if it turns, then it turns preferentially to the right. Okay? Now, second strategy. So in that case, we have only we just modulate the intensity, the total intensity of the fish, and again, we have the intensity that varies with the orientation of the fish. So if the fish points to the north, it's essentially in the dark. If the fish turns around and points to the south, then it gets a lot of light. And so we can try different profiles and do the same sort of analysis. And you can see that even though he has no stereovisual information, he's still able to point toward the sun. So not necessarily very efficiently, depending on the profile, but you still have a large bias towards the sun. So how does it do that? Because it doesn't have any information. So in that case, the only way for the animal to do something is to compare before and after. So essentially, this modulation could come from the intensity. So it could actually modulate the amount of orientation based on the intensity or the variation in intensity. And that's what is going to decide where it goes. And so to make the story short, what we found is that what the animal actually does is very simple. Whenever the light increases, it behaves exactly as if there was nothing. So if the light keeps increasing, it behaves exactly as if it was swimming around with no variation. But as soon as the light starts decreasing, which it doesn't like, what it's going to do is going to make more turns and larger turns. So it's essentially, it's going to start diffusing more. And so for people who know about bacteria, this is very reminiscent of the run and tumble process. So bacteria do chemotaxis exactly this way. They move a certain direction, they record the amount of a certain component in the water, and in that case, if the concentration increases, then they're going to keep going straight most of the time. But if the concentration decreases, then they're going to turn around and so diffuse more, and go back another direction and do that again. So essentially, that's a process that's also done with a few differences, but mostly the same way by this little vertebrate. Okay, so there's no direct bias, and any decrease in illumination increases the probability to turn and the amplitude of turning, so increase the diffusion of this little particle. Okay, so to summarize, you have a fish. The first thing you have to decide is, should I turn or should I go? And this is controlled by the change in total illumination. So if the illumination just decreased the time before, then it's going to favor turning, okay? And now, once you decided to turn, you have to pick before left and right. In fact, you have to pick before flipping or staying in the same direction. And this is controlled by the contrast. So essentially, you have two ways for the animal to control the orientation. One by the total amount of illumination, and the other one by the contrast. And they act on completely different parameters, which is, I think, quite interesting. Okay, so I think we're done. So I think we sort of understand, and we can simulate that. It's pretty easy. You can make a sort of toy model of a fish, and you can reproduce most of the distribution that I've shown you before. So we're saying that we have understood the basic algorithm, behavioral algorithm that the animal does. Another question is, can we understand how the brain produces this sort of process? Okay, so looking at the tail, when you're recording, it's not easy for technical reasons. So what I'm going to show you here is a few experiments to show you that maybe we can use the gaze, so the direction of the eyes, as the proxy for the orientation of the animal. So one thing we did, so when you want to do imaging at the same time, you have to put the fish, you have to tether the fish, you have to brog it. So what we do here is we put the fish in some agarros. You can't really see the agarros, but it's here, so it doesn't move. But it can move the tails and it can move the eyes. So you see here the position of the gaze, so the orientation, the mean orientation of the eyes. and the blue lines gives you some measurement of the tail motion. And so one thing that is really striking is the fact that the gaze and the direction of the swims are highly correlated, which means that if the animal looks to the left, there's a huge 95% chance that the bout is going to produce is also to the left. So this is something that's called coordination, which is pretty much in any vertebrate you're going to find that. In our case, that corresponds to coordination between your eyes and the head. So if you turn your eyes to the left, it's very hard to actually move your head to the right at the same time. Okay, and in fact, this correlation is also in time. There's a strong correlation in time. And so because of that, in a way, if you look at the eyes, you know the turning state of the animal. If the animal looks to the left, you know that it's in the left turning state, right? And if it looks to the right, it's in the right turning state. So maybe we can use that as a way to have some sort of external readout of the state. So in fact, if you just focus on the gaze, you see this very typical saccadic movement of the eye. So every 10 seconds, every 11 seconds, the eyes move back and forth. And so 10 seconds is also an interesting quantity. If you remember well, the animal produces a tail beat every 1 to 2 seconds, and is going to produce typically 5 tail boots to a certain direction before turning around. So 10 seconds is typically the duration of the streaks of the animal. So for how long it's going to turn to the right, and then how long it's going to turn to the left. So this alternation tells us that we may be able actually to really see this alternation between right and left state by just looking at the eyes, which is going to make things easier. So to check that if this is true, then maybe we should be able to bias the gaze itself. So to do that, what we've done is we put a fish here, we record the motion of the eyes, and we do some sort of stereovisual stimulus. So we impose either we shine light to the right or light to the left, and we can look at the distribution of the orientation of the gaze. And what you see is that if we put light to the right, then the animal tends to spend more time looking to the right. Okay? So you can actually bias not only the tail boots, but you can also bias the gaze. Okay, so I'm going to maybe go fast with this one, so we can do the same thing in retroaction to test spatial temporal sampling. And we can also bias the gaze distribution the same way. So essentially, the main point here is that what we are interested in is to understand how the animal go left or right. And now we have a way to read that, not by looking at the tail boot, but by looking at the direction of the eyes. Okay, so now the first thing you want to know is, okay, if this alternation, the saccadic alternation, which decides the navigation pattern of the animal, has to be controlled by some of the neurons of the animal brains, and we don't know which ones. We have a few ideas from the literature, but we don't know exactly where this saccadic controller might be. But now comes the beauty of the light sheet imaging and the whole brain imaging. To find these neurons, the only thing you have to do is record everything, but at the same time monitor the eyes, and then you're going to correlate the direction of the eyes and the activity of the neurons. And in that case, because you have pretty much all the neurons, then you have all the candidates neurons that might be involved in this process. Any neuron that has no correlation with this process should not be involved in this phototaxis system. So just to describe a little bit more in detail what we mean by correlation, so you take the eyes, you track the eyes, so you have the position of the eyes, you do this imaging for each voxel or neurons, you have a certain pattern of activity, you do a regression, and you get this entire map. So in that case, we went a little bit more in details. We separate the neurons that correlate with the position of the eyes and the neurons which correlate with the derivative of the position of the eyes, so the velocity of the eyes. So some neurons only respond when the eyes are moving, and then they become silent immediately. Okay, so now let's look at these different clusters of neurons. So some of them were known before. People had focused on different regions. For instance, this one is called this. Everything is in the hindbrain in that case. And these two regions correspond to the burst neurons. So they are known to project essentially directly to the motor neurons. And they have a burst of activity each time there is one saccade. So they belong to this class of velocity-tuned neurons. As you can see, each time there is a saccade, poof, you have a large activity pattern. And this one activates when the eyes go to the right. This one here activates when it goes to the left. Okay. Then there is this very interesting network here, which people have studied a lot because it's what is called the velocity position neuron integrator. So the idea is that each time this one have a burst of activity, this one do some sort of mathematical integration of the activity. So you have a direct of activity, and the other one goes above, and then they stay. And the idea is that by staying at a certain level, they force the muscle to maintain the eyes at the right position. So this one was known before too. These two clusters were not known to be correlated with the activity of the eyes. And so you can see that they pretty much follow the saccadic movement. And the blue ones activate when the animal turns to the right. When the eyes turn to the right, the left ones activate when the eyes turn to the left. Okay, so we're going to focus on this cluster, and especially on the number 3 here. And what we wanted to show is that this is the actual Saccade Command Circuit. So we think that this circuit is the one that controls the left and right alternation every 10 seconds. So why do we think that? It's because when we run experiments in which we block the eyes by paralyzing the animal or just putting agar everywhere, so you have no motion of the eyes, these two clusters tend to self-oscillate. So they have self-oscillation even in the absence of motor activity. So it's like an oscillator, a neuronal oscillator, that activates even though there is no motor output. So it looks like something people call a central pattern generator, something that can produce oscillation on its own. Okay, so, and interestingly, this self-oscillation, even in the absence of saccade, has a typical period of 20 seconds, which is twice the time between alternation. So it sort of makes sense that this will be the circuit that will spontaneously generate the saccades. Okay, so we wanted to test that a little bit more. So we went to the VR lab at the ICM, and what we did, we ran experiments in which we used Channel rhodopsin, so optogenetic, to specifically activate this region. And so when you do that, like in this movie, you can see that if we shine at the right place, we can actually impose, so when the light turns on, that means that we're actually turning the light on at this position in the brain. And you can see that we can completely control the motion of the eye. So we can take over the control of the eye dynamics. And this region in particular, so we knew that this region should activate. These are the burst neurons, but this region also, which corresponds to the cluster I was showing you before, also has a very high response, motor response, when we optogenically activate them. So it's not a direct proof, but it's again consistent with the idea that this circuit might actually be the one controlling the activity of the saccadic activity. Okay, so we have some sort of idea of this circuit. So we think that we have an oscillator, so with two parts that self-inhibit and auto-activate. This is typically how you're going to describe an oscillator, neuronal oscillator. It correlates with the eye angular position. They display self-sustained oscillation even in the absence of saccades and at the same frequency of the saccades itself. And if we optogenically activate this region, we can actually control the saccadic dynamics. Okay, so now if that is true, if this is the one controlling the gaze, and if the gaze controls the actual navigation pattern, then this one should be biased by visual stimulus, right? So what we do now is take another fish, put it in the light sheet. Now we're going to use a 2-photon light sheet just to make sure that the animal doesn't see the blue light of the standard light sheet. So it's 2-photon just because we wanted to be able to use infrared light, and the animal is completely blind to infrared. And now what we're going to do is very simple. We're going to have two LEDs on the right and the left, and so we can shine light to the right eyes or to the left eye. And we're going to select these little clusters, the blue and red. And now if I plot the activity of these two clusters, when I turn on the left diode, I see the left circuits activating and the right circuits being silent, and so forth. So again, I can take control of these circuits by alternatingly displaying light on the right and on the left. Okay, and this is the average response here. So that makes sense, of course, if we think that if the gaze goes to the right, so if we activate the right circuits, then I'm forcing the animal to go to the right, and this is what you expect from the stereo visual point of view. Okay? Now what happens if I shine uniformly some light to the animal? So in that case, some very bright, some flash, but at the same time on both eyes. So here I'm looking at one of the two circuits here. And so the first time we saw that, it's a little confusing because you get the feeling that it does respond sometimes, like here, or here, or here, right? But for some of the flash, it's completely silent, right? Okay, but now if I show you the response of the second circuit, the symmetric circuit, then it essentially fills the gap. So whenever this one doesn't respond, this one is going to respond. So you can see that the two are responding together, but there's this large variation because, in fact, they are in conflict. They don't want to be activated together. So they respond to the light by some sort of winner-takes-all situation where one of the two circuits is going to win, activate the lock, and turn off the symmetric circuit. Okay, so this is pretty hard to interpret. So you can see that the anti-correlation between the two circuits. But so to interpret that, we say, okay, maybe if we want to guess which one is going to respond, maybe we have to take into account where the system was at the time where I deliver the stimulus. So the idea is that you have some sort of oscillation, and at each time, a certain phase in this oscillation, right? For instance, if the phase is 0, that means that the left circuit is active and the right circuit is completely inactive. If the phase is pi, it's going to be the opposite. Okay, so I can sort of extract this phase by doing this technical thing called Hilbert transform. So I can measure the phase at each time. And now what I can do is plot the response of the circuit depending on the phase. So each stimulus corresponds to a different phase. And now if I regroup all the stimulus corresponding to a given phase and look at the response of the two circuits, what I can see is that the response is not stochastic completely. It depends on the state of the system when the light was delivered. And the interesting thing is that what you see here is that when the system is active on the left, which corresponds to this position in the phase space, if I give the animal some light, then in a way it stays there because I'm reinforcing the state of the system that was already there. So that sort of makes sense if you think about the system where a possibility that the animal, or the other way around, because it responds to the dark, if it goes to the dark, if it goes to the light, and the light increase, in a way, you'd better stay there. You can continue there rather than reversing. Okay, so we try to modelize that with the help of Alexis Dubreuil and Rémi Monasson at the École Normale. And so a very simple model. We have this oscillator with inhibitory, cross-inhibitory, and self-excitatory. And we have some sort of input coming from the right and left eyes, right? That is described, and that's essentially based on the data. So now, if you adjust correctly the different parameters of this model, you can produce this like stochastic self-oscillation that I've shown you before. So this circuit can self-oscillate in a certain regime. So now, in this regime, I can produce the same sort of stimulus. So of course, this circuit, if I put, if I activate just the left eye, not the right eye, I'm going to activate this one. So this is completely trivial. So the stereovisual part is trivial. The part that's not completely trivial is the fact that if I activate the two eyes at the same time, I can essentially reproduce this phase-dependent response. So also, there's nothing very complicated here. It seems that this phase-dependent response is actually something easy to produce with this sort of oscillator. So you have, essentially the thing is you have an oscillator. So at a certain point, if you shine light to the two, there's one that's more sensitive to the other and it's going to win. And it's going to win it all. And because it's going to win, it's going to turn the other one down. So that's essentially the idea. So I think it's interesting the fact that you get something that seems complicated, seems tricky to produce. And in fact, a very, very simple model like that reproduces essentially what we see. Okay, so of course, with a little model like that, you can also, put that on actual fish. So we have a fish here. We produce this oscillation, and then we have a rule, which is if the animal looks to the left, it's going to turn to the left every 1 second. So you see here, each little step corresponds to 1 bout. And when you do that, and you put some light, and you have some rules about how much light comes, reaches the eyes depending on this angle, which we make it as reasonable as possible, then statistically the animal is going to, so in that case it's 90 degrees, so it's going to peak at 90 degrees. And interestingly, it's not going to be, there is some noise, you can see. The animal doesn't really stay facing the animal because it keeps exploring. There is some fluctuation here, but there's a bias, and this bias tend to bias the distribution of orientation toward wherever the light is. Yeah?
You have one or two oscillators? You have one per eye, or you have...
No.
HVO right, HVO left?
Yeah, so there's one oscillator, which, as any oscillator, has 2 parts. Okay. So when one is active, and this is the right part, which means that's the right part of the brain, and it corresponds to the eyes being to the right. So the right part is active, or the left part is active, right? So that corresponds to these two clusters that are symmetric.
If you destroy one eye, the fish is not able to follow the light.
So we've tried with 2 eyes to make sure that it comes from the eyes, because they have like photosensitive cells everywhere. So one question that we have is this, but we didn't try with one. Yeah, that's gonna be. I'm sure that's gonna be another thing to try. I think it's so it's complicated because it's not gonna swim. They have pretty heavy eyes. So if you take away the 2 eyes, and after a few days they recover and they can actually swim pretty nicely. But I think I would be tempted to say that they're gonna be. Sometimes I won't like it.
You don't see the oscillator during the forward movement?
The thing is, in that case, we don't see the forward movement. We don't. When we record, the tail is not free. We only free the eyes, so we don't have a correlate to forward or turning movements. But that's actually we still don't understand the switch. So we understand the switch between left and right. We don't understand the switch between forward and turn. And it could be a completely different circuit. And this is something that we're looking at. So now we know to activate this circuit, we just have to decrease the overall illumination. That's exactly what we're looking at now. Okay, so I think I'm going to finish here. So just to conclude, so phototaxis and zebrafish combines plano and tropotaxis, which is just another way to say stereovisual and spatial temporal integration. So these two strategies seems to be at play in the same animal and correspond to different parameters that is able to tune during navigation. So we can use gaze orientation as a proxy for these left and right states. And as I showed you, we can do, thanks to this whole brain imaging system, we can do unbiased functional mapping. It means you don't have to guess what and where you're looking for. You just record everything, and then you pick whatever shows up, and then you can focus on that. And so that's how we identify this CPG for central pattern generator like circuit that seems to control saccades. And then the HPO, this oscillator here can be visually biased by unilateral elimination. And then we have a simple model that seems to capture a lot of this process. So here I'm showing you, the three different experiments that we did on this fish, which is looking at the neurons that correlate with the gaze, looking at neurons that self-oscillate in the absence of saccades, and looking at the neurons that respond to left and right. And in a way, that's a nice thing with this system is that you're interested in the sensorimotor process, right? So you need to be able to fuse together information from the sensory system, from the motor system, and also from the spontaneous activity. And this corresponds to these three things. And in fact, what I'm showing you here is not only one fish, it's different fish that we've recorded, and then we can fuse them together by morphing. And so you can fuse these different experiments, and then you can delineate exactly these regions where it seems to correlate with the gaze position, it self-oscillates, and it responds to visual stimulation. And that, of course, is an easy and fast way to pinpoint to the circuit that's probably of interest, in that case for phototaxis. But I think that probably is applicable to many different other situations. Okay, so just I'm going to thank, so a lot of the work has been done by Sébastien Wolf and Sylvain Carmelco that I showed you, and help for the theory part from Rémi and Alexis, and then for the development of the light sheet years ago, we had a lot of help from these people. Okay, thank you very much.